going to move away from brain imaging and talk about disrupting the brain. So here, the traditional approach would be studying patients who have lesions of the brain, and a more modern approach would be to actually kind of stimulate or disrupt the functioning of the brain, not dissimilar to the kind of Penfield experiments before, but using somewhat different methods. And here, by reverse engineering, what I mean is that you almost take something apart in order to see how it works. So by removing a part of the brain by accident or design, you can then make a conclusion that part of the brain is important for X. Okay? So it's a different kind of logic from doing brain imaging. And this would be a kind of a traditional approach of studying patients and seeing what's going on. So these are patients who have problems in producing written words, and perhaps somewhat bizarrely, you've got some patients who have problems in producing vowels and others who have problems in producing consonants. So this is somebody who's writing a word like Bologna, where they literally have lost the ability to write vowels, and it can't be that they've lost the actual motor ability. And you have what are called double dissociation. So this patient here is poor at writing vowels and good at writing consonants, and this one here has the opposite profile. What people would often conclude from this is that there are different neural resources for processing consonants and vowels in the brain, for instance. It doesn't tell us anything about where they are, so it's a different kind of logic for brain imaging. But it doesn't necessarily tell us that there are parts of the brain that are only involved in processing consonants and vowels. To do that, you have to do more experiments. So you would want to give them, for instance, numbers to write, or give them other things to draw, and then you can make conclusions to how specific they are, or whether it appears to be like that. So you can do other things like, for instance, whether or not it is just to do with motor output. So do they have the same thing with spelling words orally, for instance, so saying B-L-N-O-P, this sort of thing, for instance, to kind of understand at what level of the system it is. But either way, the idea here is that you're kind of reverse engineering the system. You figure out that somewhere in the brain there is something that is more involved in consonants, something that's more involved in vowels. They might not be uniquely involved in these things, but they're at least relatively specialized. So normally you would have two or more patients or patient groups and two or more tasks that show complementary strengths and weaknesses. And from this, we typically would infer that there are different kind of mechanisms for processing this. And this is the kind of thing that I'll talk about a lot in other lectures. So for instance, you have some patients after brain damage who have problems in perceiving colours, and others who have problems in perceiving motion. And that suggests that there are different kind of parts of the brain involved in different visual attributes. And within this, basically, you've got various ways of kind of approaching that. So the second approach here is the one that I've just talked about here, where in effect you're looking just at the pattern of cognitive performance. And here what you're trying to do is not really address questions about where things are in the brain. You're just asking how is cognition fractionated by brain damage. Where they are would be another question. What are the building blocks of cognition? Traditionally, this has tended to use single cases, although there's no reason why it has to. There are certain kind of arguments around it. The other approach, of course, is to use patients in another way. That was the example that I gave early in the lecture, for instance, of taking a group of patients where, say, damage to a particular region, like the frontal lobes, and then seeing what kind of tasks can they do with the damage that particular region. And that particular approach here does make good contact with the brain imaging, because you're using as your data a part of the brain, seeing whether it's lesion, then seeing whether or not it actually impairs that task. So remember, in brain imaging, it might be that a brain region's active, but we don't quite know why it's active. Is it kind of listening in? Is it crucially involved in the task? Is it kind of peripherally involved in the task? Whereas evidence from patients will tell you how important that region is for the task by kind of removing it. So that would be slightly different approaches. That here you're seeing the importance of a region for a task, and here you're just using patients in a more exploratory way to see how cognition will separate into different functions. An alternative approach that doesn't rely on patients is to use TMS. So TMS is a method of brain stimulation. It's non-invasive, which means that basically you're not putting anything inside the body. The magnetic field obviously goes into the brain. And let me just show you a video of that, and then I'll kind of explain how it works.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away.
Star Trek. Yeah, with very left eyebrow twitch.
It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other side of the brain.
Okay. Humpty Dumpty sat on the wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
I felt a bit apprehensive during this intriguing demonstration, but Professor Walsh thinks nothing of tinkering with his own brain for the sake of science.
Jack and Jill. That's great.
As researchers from around the world are about to gather to discuss the latest results, he told me about the remarkable potential of brain stimulation.
When we're trying to do experiments in which we establish how people might recover from stroke or some other kind of brain damage, then we can try and make very good, educated guesses about which processes we want to interfere with to help your weakened but still happening processes take over the function that you couldn't do anymore. The equipment and the things we can do with it are evolving into clinical uses now, and we have a number of problems with classical transcranial stimulation with these coils and the big machines and the mapping of the brain that you have to do when you carry out these experiments. What we're trying to think of are ways of getting brain stimulation into the clinic and into the home as well.
So the way that this works is that although fMRI's obviously got the word magnetic in it as well, they're quite different, and the fMRI is a recording method that's recording the deoxyhemoglobin. Even though you're in a big magnet, it's not triggering activity in your brain. Here the magnet is triggering activity in your brain. It's being used in different ways to do that, okay? And this is partly to do with the kind of setup in terms of how it works. So here in TMS you see this coil that's held against the head. It's kind of a figure of 8 kind of thing. Within that you've got wires that are kind of going around it. And basically what you do is that you put an electrical current very rapidly through it. So if you remember in physics, when you kind of put a current through a wire, you get a magnetic field coming off. You might remember or if you move a magnet into an electrical field, into a wire like this, you see the voltameter kind of go like that in your kind of physics experiments. And it's the same thing here, is that you've got the wire's got current in, and then it induces a magnetic field. The magnetic field goes into your brain and stimulates, in effect, electrical activity within your brain. So you've got electrical activity in the current, magnetic field, electrical activity in your brain. That's kind of how it's working. So it is kind of inducing changes within the actual neurons, causing them to fire in a way that isn't happening with fMRI, where yes, you have got a magnet there, but it's kind of an ambient magnetic field. It's not something that's rapidly changing and inducing currents in the brain. Okay. What this actually means is that if the neurons are firing as a result of the external stimulation, they cannot fire as a result of the task that they should be doing. They cannot do two things at once. So they, in effect, so you're disrupting their ability to function because they are responding instead of to the stimuli that you should be responding to, speaking words and so on, it's responding to that. The actual TMS thing field is quite focal. It's kind of about a centimetre kind of squared. But obviously what can happen is that although it stimulates that part, those neurons are talking to other parts. So you can stimulate a network indirectly, but directly you're only stimulating a small region. And because you're stimulating externally, you might stimulate facial nerves. So when you saw his eyebrows going up and like that, that was nothing to do with his brain. There will be a nerve in the scalp that is controlling the musculature in the face, and that will be stimulated as well as the brain. Okay. And it can be described as being a virtual lesion. Okay, so yeah, there are plenty of ways of stimulating your brain. It's not necessarily unhealthy. It's not used on people with epilepsy for obvious reasons, but it can be used in some therapeutic situations. So it's been used, for instance, in depression, where people have kind of lower activity in their frontal lobes, and the idea there is that over time it will kind of boost the activity by causing the neurons to fire more in effect. And there is, for instance, Cochrane reviews. So this is kind of meta-analysis of blind, double-blind studies of these kinds of clinical effects. How is it used cognitively? Well, it's used cognitively basically to try and figure out what regions of the brain are doing. So you can move the coil around so you can see what happens when you induce a current here versus there, and so on. Also, the pulse is very brief, so you've got these pulses are like 1 millisecond when you hear the kind of click. That's what's doing it. So it might be that this part of a particular part of the brain is important, but only in a particular time window. It's only important in kind of 50 to 100 milliseconds, and beyond that, it's moved on to a different part of the brain. So you can actually examine the timing of cognition as well. But in most cases, in cognitive experiments, the effects are quite subtle, and by subtle, it means that you don't get overt behaviour that you can actually see. So here you could see him kind of stuttering. That's kind of quite unusual. Normally what you have is like a change in reaction times. Instead of it taking 500 milliseconds, it will take you 550 or 600 milliseconds. So it's not something you would see with your naked eye, that you've got this thing going on, you have to measure it. But sometimes you do get observable effects, so twitches of if you put it over your motor cortex, your finger will twitch. If you put it over your visual cortex, you will get what are called phosphenes. So if you put it over the back of your brain, you will see flashes of light, for instance. With that, if you put it over a part of the back of the brain involved in vision that's involved in motion, you will see flashes of light that move. Okay? And in the opposite heavy field, and that's kind of what you've got here. So this person is seeing a flash of light kind of move down this right-hand quadrant, and I can tell you that it would be the left side of their visual cortex that would be stimulated in order to see flashes of light on the right side of space. In the dark. So how can you use this in actual kind of cognitive neuroscience experiments? So this is a study here that looks at,
So if you did it on the left side of the brain, as in she'd see the flashes of light on the right side of both eyes, so it would just go here and here?
Yes, exactly, yes, it would appear, yeah, that's right, yeah, exactly, yeah, that's this right, so it's not related to the eyes, it's related to this side of space. So with this, what they're doing is looking at how TMS over a part of visual cortex affects your ability to detect a stimulus. So here what you've got to do is find a moving cross. So you have to, the blue arrows here means that they're moving. So some things are static, some things are moving. And here your task is just to find the cross that moves. What you do is that you put TMS over a part of the brain that's involved in detecting visual movement. So that's this area here, and I'll talk about it before. It's actually the kind of area that would, under some situations, generate those, it would generate flashes of light. What you find here is that when you're asked to search for a moving cross and you disrupt a part of the brain involved in processing visual movement, that you are slower in your reaction time. So you can still see it, you're not blind to it, but you are much slower at doing it. You kind of imagine it should depend on whether the cross is on the left or the right, but they didn't analyse it in that level of detail in this study. And you actually get the same thing here when you change the experiment a little bit. So here, when you apply TMS over this region as well, but here your task is to detect a static X, but there are other X's that are moving. So here you've got kind of potential interference from these. But again, you're slower at doing that. But you might have other things. So here your task is to detect a green X, so nothing to do with movement. And here, instead of being impaired, you're actually faster at doing it. So this is puzzling, and the way that this was described is that parts of the brain involved in detecting motion and parts of the brain detecting colour are kind of in competition with each other. And by knocking out the competition, it enables you to do something more efficiently in one thing. So it's almost as if they're sending kind of noisy crosstalk to each other, and getting rid of the noisy chitter-chatter from another part of the brain is enabling you to focus on the function of the other one. So in this case, TMS was impairing your ability to do that, but it might also be that some brain regions might be kind of generating a source of noise or a source of kind of nuisance variable, and when you knock them out, you can paradoxically improve cognition. But this kind of finding is rare, and this kind of finding is the norm. But again, it might just be that we're not looking in the right place. So here we would say that the TMS of parts of the brain involved in detecting visual motion impair your ability to detect visual motion or to detect static things when other things are moving. But it enables us to address other kinds of questions as well. So for instance, what does the visual cortex of a blind person do? So if you imagine that you kind of go, you're born blind or you go blind, what happens to the back of your brain that for most sighted people is visual? So functional imaging studies with PET and fMRI show that the back part of the brain, your visual cortex, is active when blind people process sounds and touch, for instance. What we don't know is what on earth that means. So does it mean that the visual cortex is actually helping them to hear and helping them to detect touch? Or is it just that they are kind of listening in to touch signals, but they're not really boosting the function? Is it just kind of there as like an artifact or something that actually isn't helpful to the blind? So functional imaging can't actually tell you that it's important for performing a function. What you would need to do is find a blind person who damages their occipital cortex, and there are 1 or 2 cases in the world who've done this, or experimentally you can disrupt the visual cortex using methods such as this. And basically this is what you get here with TMS over the visual cortex. So here, this is the visual cortex there in purple, and these are the blind groups here. So what you've got to do here is you've got to detect a letter with your fingertip, okay? So these are Braille letters or embossed Roman letters, okay? So our normal alphabet in effect. And basically blind people here are bad at detecting tactile letters when their visual cortex is stimulated, okay? So this implies that they are using their visual cortex in order to detect and discriminate touch. And it's not just kind of activated just by chance or just by this. It's doing something. If you look at people in sighted people, this here is when you apply TMS over there, the parts of the brain involved in the motor cortex, so parts of the brain involved in the hand as well here. So what in sighted people, basically the visual cortex isn't involved so much in this tactile discrimination. It's part of the brain involved in touch that is driving a lot of the effects. And the control condition here is sham TMS. So you're not stimulating anything. You're just pretending to stimulate the brain, but you're not really. Okay. So those are the peach bars here. So here, this is like a double dissociation. If you think about the patients before, one who could do consonants and not vowels, or one could do vowels and not consonants, the same thing kind of here. Is the blind people use their visual cortex for processing these tactile things, sighted people use their a different part of the cortex there. So it's the same kind of logic that's being used here. And again, this is using TMS over time. So this is using this kind of visual search task where you've got to find, say, find a purple cross or something like this on the screen. And what you can show here is that basically there are certain time windows in these peaks here where having TMS will disrupt your performance. So this will disrupt your performance to find something that is there, and this will disrupt your performance to say that something isn't there when it isn't there. So in one case you're finding something that is there, and the other you're confirming that something isn't there. But either way, you can show that this part of the brain isn't just important, it's important at this particular point in time. Okay? So this would be your kind of baseline here. So presumably these lines are, that's the kind of baseline, and these are when that reaches. So in effect with TMS, you can use this by looking at different time windows. So you can say, right, this region is important at time 1 but not time 2. You can look at different locations. So this is the example of the blind and the sighted people. The different parts of the brain might be important for certain tasks. And in me, this part of the brain might be important. In you, that part of the brain might be important. For instance, you can give people different tasks and different stimuli. So it's a very varied tool. Lastly, I'll talk about electrical stimulation. Any more? But TMS is kind of one of the older methods. So this is a way of describing kind of brain stimulation. So in green, these are kind of our non-invasive brain stimulation. So this is single pulse TMS. So that has a very good spatial resolution. This is repeated TMS where you kind of do bang, 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 bang. This has poor temporal resolution. But there are all these methods here that are all kind of brain stimulation methods. So giving people drugs. These are the kinds of stuff you would do on animals, not in humans. But in effect, these are all part of the same family in that you're stimulating the brain rather than recording from the brain, okay? And measuring something like behavior. These sets of methods here are electrical stimulation, where you are literally passing electrical currents through the brain instead of using these kinds of more magnetic methods here. And you're either applying literally a direct or an alternating current. So this is reminiscent of kind of one flew over the cuckoo's nest type thing where you gave these kinds of electroconvulsive therapy, which is still used, but only very, very rarely. But these are very, very, very small currents, whereas those ones are very, very big currents. So it's a completely different order of magnitude, but it is technically the same kind of principle. And it is done safely in labs to kind of study the brain basis of cognition. And this is the kind of thing that you would have, is that you would have two electrodes on the brain, and here these are recording electrical activity. They are passing a current through your head. Okay, so it's completely different from EEG, where it's just recording. You are doing something like this. And in direct current, you've got an anode and a cathode, and basically you've just got, what that means is the direction of the current is flowing from one direction to the other. Sometimes you would put the one that you're not interested on another part of the body, like the shoulder. So you might have one electrode on the head, and the other would in effect be your control electrode that isn't going anywhere of interest to you, for instance. In this example, it's two parts of the why would you do this? Well, actually what happens is that these, depending on the direction of the current flow through the brain, you will either stimulate the brain or you will suppress it. And anodal, in fact, what it does is that it increases the activity of the brain, and cathodal does the opposite. So depending on whether you put the anode or cathode on the right region, will do that. So here this is looking at EEG measures after stimulating the brain. So you would just put these electrodes on the brain for 20 minutes, switch them off, and then you would show flashes of light and see how the brain responds. Okay? So it's two different phases. You stimulate the brain, and then you record from the brain and see what's different. What you find is that during anodal stimulation or after anodal stimulation, you've got an enhanced neural signature. So this is with EEG to showing visual stimuli, and that's what you see here. Is that anodal stimuli will increase the excitability of the visual cortex to light, whereas cathodal will do the opposite. It will make them less excitable. So what does this mean? It means that you can use this electrical stimulation two modes. One is a little bit like a virtual lesion, a little bit like the TMS, where you're disrupting performance. But the other you can actually use more as kind of a cognitive enhancer. And this is actually being used, for instance, to enhance people after they've had stroke, to make their motor cortex more excitable. It's used in depression, and it's also even been used in children that they apply these electrical stimulation when they're doing things like training people who've had dyslexia or problems with numbers. It's slightly bizarre that this is happening, but this is happening, that you are doing this with kids to improve their reading and numeracy. That you apply this stimulating anodal electrical current whilst they're engaging in cognitive tasks in order to enhance them. Okay. And this, the idea is that at the moment this is stimulating or depressing electrical activity and changing the actual plasticity. So it's actually creating longer lasting effects afterwards. So what happens is that it makes them electrically active, and then if you do a useful task, then they settle on a useful cognitive pattern in effect. So any questions about electrical currents?
How long do these effects last? Like, would you have to keep doing it? Or?
Yes, you do. So basically, if you were to do it experimentally, so this kind of study here, they would go within 20 minutes.
Okay.
But if you're doing it as any kind of therapy, you would do it daily.
Okay.
For that. And the idea there is that basically it's not that your brain will then be continuously electrically active, it's just when it's electrically active, you can then cause it to generate more synapses during that actual phase, and that is the long-lasting effect. So the electrical activity, even when you're dealing with children or brain damaged patients, it's not that that part of the brain's always active. The electrical activity just enables you to then kind of boost the synapses to join together, or if you do it the other way, it would stop the synapses